All right, welcome everyone. How is everybody doing? Doing great? Awesome. So one more reminder to please fill out the first day survey before Friday, because what we're going to do Friday is try to coalesce together into teams and, start meeting each other, etc. So the better you know your classmates and the better they know you, the better. So if you could fill it out by hopefully Wednesday or Thursday at the latest, then it'll give us enough time to organize everyone, coalesce, and then start creating teams for the mentoring session on Friday. So on Friday is one of the formal mentoring sessions. So in addition to the first day survey, we're going to ask you to sort of complete one-page profile, and we're going to give you more information about that. But basically for now, just remember to upload your profile. All right, so here we are with Lecture 2, Expression Analysis. So here's where we are. So we are now entering the first module on genomics, epigenomics, single-cell networks, and circuitry. Today, we're going to start with expression analysis, which is going to be fairly, sorry. I don't know how to hide the captions. Do you guys see captions somewhere? Okay, I think if I hit Alt, they should go away. No. Okay. Anyway, I apologize for. Oh, no, this is actually a feature. It's for those who cannot hear me. There's always a text. We're trying to be inclusive here. Okay, so I apologize. It's hiding part of the text screen. Okay, so the goal of module 1 is to basically first, today we're going to look at expression analysis, and we're going to introduce the concept of clustering and classification more broadly in the context of supervised and unsupervised learning. And we're going to sort of include some ideas of semi-supervised learning and so on and so forth. We're going to first introduce a very simple algorithm known as k-means, and then we're going to see how this is in fact about Gaussian mixture models. And we're going to sort of, get into the mood for machine learning and Bayesian inference and generation versus discrimination, etc. On Thursday, we're going to talk about sequential data and an alignment, dynamic programming, hidden Markov models, parsing, posterior decoding, HMM architectures. Next week, we're going to get into gene regulation. So we're going to talk about regulatory genomics and epigenomics. How do we understand the motifs of gene regulation? How do we model the signal of histone modifications, etc.? Then we're going to have a formal lecture on all of the intricacies of single-cell genomics. Today, we're going to see a very simple version of that focuses on the pure machine learning, clustering, classification components. But then there's a lot more complexity, and that we're going to cover in a dedicated lecture that talks about both single-cell genomics, but also spatial transcriptomics and next-generation technologies, and also nonlinear embeddings that we're going to be visualizing today, but really diving into the methods of in a few weeks. And lastly, we're going to talk about graphs, principal component analysis, dimensionality reduction, regulatory circuitry, and so on and so forth. So that's the goal for module 1, which we're starting today. For module 2, we're going to be looking at protein structure, module 3, chemistry, module 4, EHRs, etc. Sounds good? Excited about module 1? So again, this course is called Machine Learning in Computational Biology. So today is going to be our first formal introduction to machine learning. So, first, what is machine learning? Machine learning is the ability to improve on a task with more training data. Okay? A lot of people have been talking about AI since the 60s. They're like, oh, artificial intelligence. Artificial intelligence is not necessarily machine learning. Artificial intelligence could be, oh, I'm going to code in a bunch of cases and decisions that the machine is going to make so that it looks intelligent. But if that machine is not getting better with more data, it's not actually machine learning. Is everybody with me here? So that's the key idea. First, there's a task. Then it's about training data. And it's improving. Sounds good? Awesome. So what is that task? That task could be many, many different things. The two tasks that we're going to be talking about today is classification and also clustering. Classification is a form of supervised learning. That's when we know the answer. When you know the answer, then there's somebody supervising the algorithm. When you don't know the answer, it's unsupervised learning. So basically, one task could be, how well can I classify Alzheimer's patients from controls? With more data, if I'm getting better, that's a type of learning. Everybody with me here? Another task could be regression. I want to predict the age at which a person will get Alzheimer's. Regression is when I'm predicting a number, not a class. Everybody with me here? Transcription could be simply taking a voice stream and doing what you see on the screen here that I couldn't get rid of. Translation could be doing it in, a bunch of other languages like Greek, French, Spanish, you name it. This could be structured output, namely, I could speak and it could be, for example, I could be speaking to a patient about their diagnosis, and the machine could be creating structured data from this. So when we talk about structured versus unstructured data is usually free text or an image. Structured data is, tumor size 7. The image is just a bunch of pixels. It's unstructured. Tumor size 7, it's something that you have inferred from the image. Everybody with me here? Diabetes stage severe, that's structured data. Unstructured data is, well, you're starting to show symptoms of cognitive decline, we're a little concerned, etc. Okay, that's unstructured data, free text. Everybody with me here? Now, machine learning used to be very much about structured data. Basically, you have a bunch of variables and you're doing stuff with those variables. It's about data, it's about numbers. I'll show you some slides where that dichotomy between structured and unstructured data is starting to get lost, where you can basically start doing clustering, as we're going to see today, but now on documents, on text, on patient records or patient responses. Okay, so it could also be anomaly detection. For example, you have a bunch of streams, and at some point you want to raise the alarm, like, oh, the patient has fallen down. So basically, Dina Katabi upstairs, she's basically modifying Wi-Fi to be able to sort of observe people walking in their homes. And there's going to be all kinds of activities. At some point, you're going to have to make a detection to say, okay, call the cops, call the medical team. So that's anomaly detection. Synthesis is about taking a lot of data sets and then summarizing them better, sort of inferring something about the field. Imputation means finding missing data and then filling it in. And then denoising means finding sources of variation that are not necessarily meaningful, that are non-biological, that are simply, perhaps technical, and so on and so forth. Everybody with me here? So in all of these tasks, through the course, we're going to see techniques that allow you to get better with more data. And that's going to be, what we call machine learning. And then when we say improve on a task, that basically means we need to have some kind of performance measure that tells us that indeed we are improving. Everybody on board? Good. Questions? Okay. So now when we talk about training data, this is what we're going to call the experience. Okay. And then the experience is either the robot running around in the world or the AI agent monitoring interactions with patients or figuring out how the doctor is responding to different circumstances, getting better at this. Okay. So that's basically our working definition of machine learning. Okay. Good. Now, we're always talking about making inferences about the world. And in computational biology, there's always going to be something that is observed. And those are going to be the expression values, for example, for a gene. And then there's something that's going to be hidden. And that's, the patient has Alzheimer's. And we're going to distinguish these two different worlds. There's the world of the measurements, and then there's the world of the model. And inferences is inferring something about the hidden state of a system based on observations about that system. Okay? So what are the observations? For example, if you're looking outside, I don't know, some long hole in a wall, and you can see that there's a lot of bright light, you might infer that it's, summertime when you woke up, for example. Or if you see, a lot of gray, you might infer that there's, rain, or that it's fall based on the observation of rain. So basically, when you see snow outside, it's unmistakable, it's snow. it's rain, it's cloudy, it's sun. This is the observations. The inferences is, what season it is, or what is the, storm system that is going on above me. And I have isolated observations, but from those observations, I'm inferring something about the hidden state of the world. Why do we care about that? Because I can then make additional predictions. I can basically say, oh, perhaps I shouldn't go sailing because there's a storm building up. Okay? So that's the whole point, that you're observing parts of the world, and then you're inferring something about that world, about something about the underlying hidden drivers of those observations. So this is the world of observations, of data, of measurements, and this is the world of models, of hypotheses, of inferences. Everybody with me here so far? Great. So very often it's going to be easy to express the forward probability of an event. That basically says when it's winter, then there's, 60% chance of snow. When it's summer, there's, 60% chance of sun. Okay? So these are the forward probabilities. And this is typically the way that we express systems. Why? Because they're easy to measure. You're basically saying, okay, now it's summer, I can measure a bunch of stuff, and then this is what I find. Or I have an Alzheimer's patient, I can measure their expression patterns. This is the probability of having that gene expressed highly when you have Alzheimer's. Inferences are going to be about reversing that directionality of the arrow. It's basically saying, given that I observe rain, what is the chance that there's a storm system above me? Okay? Given that I observe high expression of, some lipoprotein gene, what is the probability of Alzheimer's? Okay? So very often we're going to be talking about the forward probabilities, and that's the generative probability. In other words, I am in a state of the world, for example, I'm in the summer state, and I'm sampling observations from that state. Based on knowledge that it's summer and a series of observations, I can infer the probabilities of that generative model generating samples from a distribution. So for example, I'm sampling temperature readings from, June, and then I get a distribution of temperature readings. Now, given that I have a given reading, I can say, where does that reading fall in that distribution? And from the reading and the underlying model that I have of, say, the standard deviation and the mean, I can say, okay, well, the June state generated 72 degrees with, this probability. The July state generated 72 degrees with a different probability. And now I can start making inferences about, is it more likely to be June or July based on the measurement that I have. Is everybody with me? I mean, this is a terrible example because this is Boston, so it doesn't actually work that way. Boston basically just shuffles all the days, just randomly pulls out of weather. And then, imagine we're in some other sort of well-behaved climate zone. Okay, everybody with me here? Awesome. So basically, when we talk about a generative model, we're basically talking about the sampling probabilities from an underlying model that is able to generate observations of the world. Okay? And therefore, we have the probability of an observation given the season, or the probability of snow given winter, or in general, probability of a measurement given a state of the world. And what we're trying to do is reverse the arrows and basically make an inference about what is the probability that the patient has Alzheimer's given a bunch of gene expression measurements. Okay? So basically, we have the probability of the data given the hypothesis, or the state of the world, if you wish, and then we're trying to infer the probability of the state of the world, i.e. patient has Alzheimer's, from the observations, i.e. I measured, this expression for that gene in that tissue. Okay? And that's where Bayes' rule comes in. So basically, when we talk about Bayesian inference, it sounds fancy, but it's just simply, we're using Bayes' rule to reverse this arrow. We have the arrow of probability of hypothesis given data, and then we're flipping it around by talking about the probability of data given the hypothesis. hypothesis given data is like given data hypothesis. So the arrow is pointing down. Data given hypothesis is, sorry, that's the arrow pointing down, data given hypothesis. So we have hypothesis, we generate data. So that's the arrow pointing, given hypothesis data. And then this is given data hypothesis. In other words, we're trying to infer the up pointing arrow. Okay. And this is super simple. It's basically simply the probability of a hypothesis given the data is the data given the hypothesis times the probability of the hypothesis over all the data divided by the probability of the data over all the hypotheses. Okay. To explain that super simply, it's, the purple area is simply the fraction of the world that's red times the fraction of the red ones that are also blue. But the purple area is also the fraction of the world that's blue times the fraction of the blue that's also red. In other words, P of A given B times P of B. In other words, P of B is the fraction that's blue. And then P of A given B is the subset of that that's red. P of B given A given times P of A is like simply P of A, that's red. And then this is the part of red that's also blue, purple. Everybody with me here? So that's simple to derive. And then, from that, you get P of A given B equals this divided by B, and that divided by A, okay? And that, in the context of Bayesian inference, is simply that the posterior probability, what does that mean? Why posterior? Because I've seen the data. So post means after. So after seeing the data, then the probability of my hypothesis is something, okay? So having seen snow, my probability of winter is something. Everybody with me here? And then having seen the expression of, a gene, my probability of Alzheimer's is something. And I can infer that from simply what is the probability of expression of that gene given Alzheimer's. But notice I have to worry about the prior for Alzheimer's. Basically, very, very often, people confuse those two and they're like, oh yeah, well, if this is higher, of course, this is higher. And yes, it's true, but for hypotheses that have vastly different priors, then this is absolutely not true, okay? In other words, I take a test for some rare disease. The test comes back positive. What's more likely, that I have the disease or that I don't have the disease? It's still more likely that I don't have the disease. And the reason is that the probability of having a rare disease is like, I don't know, 1 in a million. A given rare disease. Having some rare disease is much higher because there's tons of rare diseases. But having a given rare disease, that probability is extremely high. That's why we don't go around testing people for everything. The doctors just don't go, testing for everything because these tests are fallible. They basically wait to have some hypothesis that you might actually have the disease based on some symptoms, and then they do the test for that disease. Everybody with me here? So basically that prior is extremely important when you start thinking about, rare events. For example, having Alzheimer's or, the test might come back positive, but the prior might be so small, okay? So that's why we need to worry in Bayesian inference very much about not just the likelihood, that's the probability of collecting the data when the hypothesis is true. In other words, seeing something given that I have Alzheimer's, but also the prior, which is the probability that the hypothesis is true before collecting the data. And we call it prior because it's before the data, posterior after the data, okay? Now this bottom here is the marginal. This is simply the probability of collecting data under all possible hypotheses. So what's P of D? P of D is P of D given H1 times P of H1 plus P of D given H2 times P of H2 plus P of D given H3 times P of H3, etc. So this is the integral over all possible hypotheses. Does that change the most likely hypothesis, the most likely model? Basically, for example, if I'm worried between Alzheimer's, schizophrenia, or bipolar for a patient, does that change anything? No, it doesn't. The only thing that matters is the prior when I'm comparing between different hypotheses, because that marginal is going to be the same for all of them. Okay, the total likelihood, the total probability is going to be the same for all of them. Everybody with me here? Awesome. Any questions? Okay. So in that context of basically, Bayesian inference, we're going to be first introducing this very simple algorithm for clustering. And then we're going to basically see that it's just a simplification of a much more general algorithm for Gaussian mixture models. What are Gaussian mixture models? It's basically when you have a mixture of multiple Gaussians generating data. So I'm sampling data from the green distribution, I'm sampling data from the red distribution, I'm sampling data from the blue distribution, and then I observe a bunch of data points. That's the actual process generating the data, but I don't know that process in advance. What I need to do is figure out what is the number of clusters, where are the centroids of these clusters, what are the spreads and distributions for each of these clusters, etc. Okay, so unsupervised learning is going to be about, without any guidance as to what's green, red, or blue, inferring what is the underlying distribution that might be generating my data. And that distribution might sometimes be a mixture of Gaussians. Everybody with me here? Awesome. So this is unsupervised learning. And then supervised learning basically says, oh, I've sampled a bunch of green points, I sampled a bunch of red points, I know the labels for those points, I get a new point, what is it? Okay. So if I get a gray point somewhere around here, you're going to guess red. If I get a gray point somewhere around there, you're going to guess green. If I get a gray point somewhere around there, you're going to be like, well, you know, it's hard to tell. Everybody with me here? So basically, these are the two main tasks that we're going to be talking about, clustering and classification. So basically, clustering is here, classification is here. And in a toy example, we're going to be, you know, thinking about expression, for example, in the brain or expression in the liver, and the, you know, single dimensionality projection of this is going to give you some perhaps separable data, but projecting on, for example, liver, there might be some overlap. But if you look at both dimensions together, the data become more separable. Everybody with me here? Awesome. So we're going to be talking about a set of objects that are characterized by one or more features. The features are going to be all of the measurements we have. And when we talk about 20,000 dimensional objects, it's simply that we did 20,000 measurements. For example, all of the genes in the human genome. It's routine to just measure all 20,000 genes. And then you have 20,000 features. Now, how well can I classify every person in the room using 20,000 features? Super well. You know, I can figure out exactly who's wearing a blue shirt or a pink shirt or a, you know, I don't know, green earrings or you name it. Why? Because I have 20,000 features. Super easy, right? So basically, we're going to be worrying a lot about, do I have too many features? Do I have way more features than objects? So if I, you know, if I have, I don't know, 70 students and 20,000 features, super trivial. You know, I can certainly find a gene that classifies any one person in the room. Okay, yeah.
When we have so many features and we want to visualize it, do we want to use something like PCA in order to do that?
Very, very good point. So I'm going to be showing you plots that are usually two-dimensional because, you know, we're human and we're having trouble visualizing more than two, certainly more than 3 dimensions. So most of the time, I'm going to be showing you some lower dimensional projection of these very high dimensional data. And I'm going to show you some of those. And when we get to the corresponding chapter, lectures, probably lecture 7, we're going to be talking a lot more about how those are made. Now, PCA's principal component analysis basically says, what are the major components of variation? If you look at here, the major component of variation is this one. And the next one is that one. Now, I went and measured expression of, let's say, this gene in brain and that gene in liver. Is that the right dimensionality? Or is the right dimensionality some underlying process, for example, lipid metabolism overexpression in that person, that then gets reflected both in the expression in the brain and in the expression in the liver? So maybe that principal component, maybe that combination of features is actually where the true biology lies. And then what I'm seeing is different projections, which are only capturing partial information about the true underlying biology. Is everybody with me here? So sometimes these axes of variation are going to be interpretable, and it's going to be, oh, simply the combination of those two features. Other times, the global axis of variation will be completely meaningless because we've taken 20,000 dimensions and we compressed them down to 2. But then the local variation will still be very meaningful, at least biologically. For example, as you're moving in one direction, you might see that, oh, those cells are more Alzheimer's and those cells are more healthy. That dimension absolutely means something. But it is reflected in perhaps 3,000 measurements at the same time. because all of these genes are slowly increasing their expression along with Alzheimer's. But the true biological variable is Alzheimer's. Does that make sense? So basically, we're going to be talking about both linear and non-linear dimensionality reduction. And the linear one is when I do PCA, and the non-linear is, for example, t-SNE and UMAP and other sort of projections. Yeah.
So if you do like an unsupervised learning method like clustering and you see really strong clusters, can you turn that into like a classifier, or like, can you start adding your own labels to these things? Or
Yeah, validation? So if I see this data and I say, oh, well, that's class 1, that's class 2, that's class 3, and then I say, okay, how well can I classify those 3 classes? I'm going to be doing really great because I define the classes based on my clustering. And if I say, oh, this point was labeled Alzheimer's, but it's probably a mistake, let me relabel it control. I'm going to do a little data cleanup. My classification is amazing. So you have to be very cautious about that in the sense that statistically, if you basically first look at the data, there's a whole slew of things that you can no longer do because you've, dirtied your mind by looking at the data. You suddenly are soiled because you can't think unbiased anymore. So that's going to be one challenge. However, you could say, well, I'm going to do PCA and then, find the principal components of the data. That's an unbiased operation. You don't need to, it won't change based on what you see. But the moment you say, oh, and I found 10 principal components best explain the difference between Alzheimer's and non-Alzheimer's, that's biased. If instead you say, I find that 10 principal components explain the most relative variation, and then there's diminishing returns after that, and therefore I'm going to stop after 10 principal components before looking at the Alzheimer's classification, then you're good. So basically, you have to constantly be thinking, how much did I bias the analysis because I peeked at the result beforehand? So, however, there's a whole class of semi-supervised learning, where you basically have some points that are labeled, some points that are not labeled, and therefore, instead of having all points that are green or red here, and then a gray point shows up, you have a ton of gray points and maybe 3 red labels and 2 green labels, etc. So that basically means that you have to somehow use the structure of the data to understand the overall patterns of variation. And then after that, you can kind of layer in the classification component. And now you can kind of use both the structure and that. There's also self-supervised learning. What is self-supervised learning? Self-supervised learning is a lot what we see with AI right now. I talked about it in the first lecture, where we basically have, billions of images and no time to annotate them all. So I'm going to hide a patch of the image, and I'm going to ask, hey, what's hiding in this patch? Or I'm going to turn the image black and white and say, hey, what was the color? I have the answer because it came with color. But now the machine doesn't have the answer because I only gave it the black and white. So for the machine to figure out how to color it, it better understand what our world looks like and therefore build underlying models of the world. But now I have as much labeled data as I want for free because I just remove the color and I'm asking for the color again. This is known as a pretext task. I don't care about that task. I have the color. I hid it. So I don't care for the answer. You don't have to get really good at it. But getting really good at it means that in doing so, you're learning about the state of the world. So semi-supervised learning and self-supervised learning kind of blur the boundaries between supervised and unsupervised. These are fantastic questions. Any other questions? Okay, great. So classification, you have labels, and you want some kind of rule that will accurately assign labels to new points. Existing points, so easy. New points is where, the rubber hits the road. And the subproblem very often is feature selection. Namely, maybe there's a bunch of extraneous features. Maybe those two are perfectly correlated with each other. Maybe I don't need both. Maybe the true biology is here. Maybe I have to sort of construct, some inferred features from the measurements that I have. Those inferred features could be some kind of linear or non-linear combination of those features. For example, this plus that, or this times that, or this squared divided by that cubed, or you name it. Okay, so this is, sort of feature augmentation, and that allows you to sometimes find dimensions through these combinations of your existing measurements where the data becomes linearly separable. And that's what support vector machines are all about. It's like that's what the kernel construction stuff comes in. It's basically a fancy form of not feature selection, but feature inference, feature construction. Okay. And when I talked about representation learning in the first lecture, or I should say call it lecture 0, basically in the intro lecture, I talked about representation learning. Representation learning is really about feature inference. So you have a bunch of pixels, but you're inferring meaningful features. And those meaningful features are closer to the underlying biology, to the causality, to the mechanisms than the raw measurements. Okay? So very often we're going to care to infer the true dimensionality of the data, the true axis of variation. And maybe instead of having brain expression, liver expression of X or Y gene, maybe the axis of variation would be male, female, Alzheimer's control, young, old, African American, European American, and so on and so forth. Is everybody with me here? So these might be the true dimensions of variation of your data. And if you can understand enough of the structure of the data to understand these latent dimensions of variation, unsupervised, and then ask, hey, what did they mean? And then you're like, oh, well, this correlated with age or with sex, etc. Then you're doing really great. There's an alternative, which is you can start with age and then say, okay, what correlates with age? Let me now take that component out. Okay, what's correlating with sex, biological sex? Let me take that component out. What's correlating with, disease status? Let me take that component out. And you can progressively peel components of variation, and then you can do an unsupervised clustering of the rest, or you name it. Everybody with me here? Cool. Other questions? Okay. So again, classification has some labels. There's some kind of feature selection, and the metric, you can actually have a metric because you know what's green and what's red. You can hide the label at this point, present it to the model as gray, and say, how well did I reconstruct it? What is my false positive? What's my false negative rate, etc. Clustering has no labels, and it's very hard to sort of have a metric for how well are you clustering. So the goal is to group points into some clusters based on how near, how proximal they are to each other, and to identify some kind of structure in my data. And the metric is usually I'm going to bring in some other measurement that I haven't shown to the algorithm, and I'm going to ask it, how well are you capturing that measurement? Of course, if that measurement is highly correlated with one of the measurements that I already have, that's a problem. Basically, usually I want to have some kind of independent validation. That's not just like maybe, it could be, for example, an imaging data set, or it could be a performance in a different test or something that was not used in the deciding of what's Alzheimer's. Okay. Awesome. So again, this is the first lecture technically. So I think it's important to sort of distinguish all of these different types of things. So basically, we've talked about AI, we've talked about machine learning, we've talked about deep learning, and I can't try to introduce them at the beginning. So basically, AI encompasses many things, including systems that pretend to be smart, where everything's hard coded. For example, a knowledge base or a rule-based, robot. That's still artificial intelligence. Okay. Within that is machine learning. And that's basically when I'm getting better at a task with more data. Okay. So for example, AI could be simply, have some input. I have some hand designed program that decides what to do based on that input. And then I have an output. The learning part are the gray boxes here. Okay. With classical machine learning, you basically have designed by hand some features. These are the measurements that you're making. And then you're inferring the parameters with which you weigh these features. And that's the learning part. The more data you have, the better that weighting will be. Everybody with me here? Conversely, if I, not conversely, further, within machine learning, there's representation learning. And that's basically when I don't design the features by hand to start with. I don't say, oh, expression of this gene in brain is, a good feature for Alzheimer's. Instead, I say, go figure out the features. Okay. And then from the go figure out the features, it could basically do some kind of principal component analysis, or it could do all kinds of other things to infer what are good features to even use in inferring the output. And I can learn the features just based on the input. I could basically say, great, what are components of variation? Or I could learn the features that give me a better classification for the output. Okay, so I could basically say, oh, let me infer some features, map the weights of those features, and then infer the output, and now backpropagate how to improve the features. And then try again, update the weights. I get a slightly better classification. I go back and I update the features again, okay? So the concept of learning, not just the weights, but the features themselves, that's what representation learning is about. Is everybody with me here? Yeah.
What would that look like for something like expression data, like RNA-seq data, for example? Would it be hand designed features if you give it like the actual expression values, or if in a representation model, what would you have to give it instead?
Yeah, that's exactly right. So basically the vanilla sort of representation learning approach would be, okay, let me now infer like some linear combination of features or some nonlinear combination of features. And then I can see every pair of features. That could be a representation. The difference with deep learning is that these features are not built at just one level. They're built on each other. So basically, I'm learning some simple features. From these features, I then learn more complex features. From the more complex features, I then learn even more complex features. Basically, having multiple layers of combinations of gene expression values is, where this would be, okay? And that's why we call it deep, because you're learning multiple layers of more and more abstract features. Everybody with me here? Awesome. Basically, that's sort of the hierarchy that we're going to see. And then within deep learning, there's generative AI, et cetera. So basically, there's, more classes that are being constructed. Who feels that they've learned stuff? Yes? Awesome. All right, let's dive right in. So with classical AI, the way that machines were kind of learning how to play chess was that the human would program all of the rules of chess. It would the human would handcraft a scoring function for every position. And, the machine would then search the moves that it can make for the whites to maximize the score, then for the black to minimize the score, then for the white to maximize the score again, for the black to minimize the score. And that's sort of the minimax algorithm that basically says at every step, I will choose the thing that maximizes my score, and my opponent will choose the thing that minimizes my score, and I will choose the thing that maximizes my score, and so on and so forth. Okay. And this is Garry Kasparov. So I met him in Croatia. He's a really awesome guy. Our kids the same age, and we also play chess. I will not tell you who won. This is an exercise that they read. So basically, the machine basically infers from the rules that the human made, and perhaps from some scoring function that the human made. I don't know how many of you guys made a chess game in your youth, but there was a time that we would basically say, oh, what is this? Is the best scoring function for where the knight is? And based on the knight being in the middle, it has more controlling squares, and therefore it has a higher value. And I can use that to score every position. And humans would basically come up with awesome scoring functions. And then the machines would basically just run through these really, really fast, run the minimax algorithm. And then that's where Kasparov lost to a machine back in '99 or something like that. And then the world was shaken. At the time, we thought that humans would be unbeatable at this kind of thing. And eventually, machines got better and better and better. And, anyway, I'm not going to get into what's happening now, but, needless to say, a lot has changed. So this was the classical way that humans would play chess. And then based on this, we had this concept of, things that are easy for humans and things that are easy for machines, okay, or hard for machines. And the things that were hard for humans were things like chess or, math integration or remembering 3,000 things or reasoning and, planning and logic, etc. But those, it turns out, were kind of easy for machines. You could basically start, planning out a path by just simply, doing depth first search or breadth first search and then sort of trying every possibility. You could, play chess by simply testing every possible option. And then there was this dichotomy of things that were basically very easy for humans that, we didn't think of as intelligent. And then, the intelligent part was things like playing chess, okay? But it turns out that the classical AI stuff was basically, I'm sorry to be so provocative, but things that humans had to get trained at. In other words, math integration, nobody's born knowing how to do integration. You know, we kind of go through school to figure out how to do this. Nobody's born knowing how to be a good doctor. No, you learn how to follow flowcharts, and that's what makes you an awesome doctor. You basically look at that evidence. Oh, you forgot that one. You're not as good of a doctor. Okay. And guess what? That's not what humans are best at. That's what machines are best at. So we trained humans to be machine-like in a bunch of tasks. And those were the hard tasks at the time, because it took us many years of education to learn how to do something properly. But machines, super easy. That's what they do best. Where deep learning is going and where the modern movement of generative AI is going is kind of like the opposite. The stuff that humans are kind of born knowing how to do. Like, you know, you're born knowing how to drive a car. Well, maybe not. But anyway, you're born to learn how to recognize images. Babies can recognize images even before their retina is like fully functional and their visual cortex is fully formed. Like, you can recognize patterns just, you know, from just being a baby. It doesn't take any special skills. You can recognize people. That's what we do best. And that turned out to be super, super hard for the machine. But it's kind of embarrassing because the machines are now getting super, super good at these things. And maybe general intelligence lies somewhere in that direction. My guess is that it lies somewhere in both directions. So basically right now, the movement is very much towards, you know, sort of large language models that, you know, have enough parameters to understand the world. But I think there might be some room for learning the laws of physics and, you know, having some kind of equation and some kind of structured modeling of knowledge and knowledge graphs and stuff like that. Maybe. But again, we'll find out. We don't know where AGI is. My guess is that it's already here. And, you know, we're just not calling it that because we're protecting our own, I don't know, vanity or confidence. Anyway, but again, if AGI is already here, then we don't have to worry about it. It hasn't killed us all, right? So you know, there's a good way of seeing it. Everybody with me here? Awesome. So again, sorry for taking the time to sort of explain this concept, but they're going to be hopefully helpful for thinking and conceptualizing about this. So when we talk about deep learning, we basically mean that there's layers of abstraction building on features on features on features. And that's basically what's being done inside our deep learning apparatus that we call our brain. Okay. So basically, we have layers in our cortex, and then individual neurons are sort of sensing perhaps pixels, and then they're sending signals. And now you can arrange neurons in a pattern that will recognize, you know, low activation, high activation, low activation. You can hard code that in your brain. And then when you see an edge, the neuron that recognizes that pattern will fire. And that's one layer of abstraction. And now that you have an edge recognizer, you can kind of build, you know, a recognizer of triangles and so on and so forth based on those features. So basically the concept that you can learn from the building blocks more and more abstract items is what, you know, is at the foundation of deep learning. We'll talk a lot more about that when we get to image analysis. And we've basically expanded this hardware, like our actual cognitive hardware, dramatically since, you know, divergence from the chimp of a few million years ago. So basically, humans have expanded this neocortex, which was already insanely enlarged, you know, from mammals to primates, and you know, has just gotten wild. And again, if you look at most humans, you kind of think of the eyes as the top of the face, but if you've taken a drawing class, you realize that the eyes are actually the middle of your face, and we just have some hair here. But when you see an extraordinarily smart human without hair, you kind of realize that the eyes are kind of in the middle. Okay, but all of you have the same thing. It's just you have hair. All right. So basically, that architecture was basically utilized in the visual cortex. But guess what? We studied the motor cortex. Nearly identical architecture. You study the, you know, all of these sensory cortices are nearly identical in architecture. Receive signal, abstract it, and then, you know, boom, you get something out of that. And then the last frontier is the prefrontal cortex, basically where all of your cognition and thinking happens. And guess what? That's the same architecture. So basically, this concept of abstractions and reasoning, you know, might be inherent to sort of how humans think and function. And this is what I was describing in, you know, the introductory lecture when I was talking about these representation learning. Okay, maybe all of these layers are just really good at abstraction, at sort of learning more and more complex inferences about the world. So we have the world of data, we have the label, the why, the prediction, and then this is where the magic happens. This is where the latent spaces are. Okay, so again, what we just saw will apply everywhere. Okay, so yes, we are in the expression analysis lecture, but it's also the intro to machine learning, intro to sort of all of this, wealth of both techniques, but also ways of thinking about the world. Everybody with me here? Awesome. All right, so now let's basically see how we can use machine learning for gene expression analysis. How can we learn? How can we get better at a task with more data? First, we're going to look at, gene expression analysis in general. Then we're going to look at this Gaussian mixture model of, I think it's a very nice foundation to start thinking about machine learning and inference. And then we're going to sort of look at actually an easier version of k-means clustering. But let me dive right in. So first of all, what is gene expression analysis? You basically start with a tissue. You then infer where, for example, the nuclei are, and that's usually much easier to capture in a tissue as complex as brain. You extract nuclei, you package up nuclei in individual bubbles that have the same barcode for all of the cells in that bubble. And therefore, all of the RNA molecules inside that one cell in that one bubble have the exact same barcode. You then sequence all of the reads that come out. And what you get, what would get back is barcodes for individual cells that I'm gonna now call the green cell. Of course, there's no color, it's just barcode GGA, TTTDC, ABC or something, or AGC, definitely not B. And from that, you will say, oh, all of these reads came from the same cell. Is everybody with me here? And therefore, in the end, you're going to have all of the measurements that you saw an RNA molecule from gene 2 in the same cell. I'm just going to number them arbitrarily, cell 1, cell 2, cell 3. And you're going to get an expression matrix, which is simply the number of times I saw an RNA molecule mapping to that region of the genome that corresponds to this gene with a barcode corresponding to cell 1. Okay? It used to be that it was expensive to sequence. And every letter that you read from the DNA cost $1. And to sequence the 3 billion letters of our DNA, it took $3 billion. Right now, it takes about $300. So we're talking about 7 orders of magnitude, from 1 billion to 100. Okay, just, and therefore, instead of just hybridizing a read to a region with a probe, we just resequence. Whatever, just resequence. Okay, and that's basically how we do quantitative expression analysis. We're basically just simply tagging with DNA barcodes, individual cells, and then measuring, and you get back a matrix. Everybody with me here? And then you basically have a number of, like you have 20,000 human genes, maybe, I don't know, 4 million cells, and you get a matrix like that. And from that, you can do your principal component analysis, et cetera. And you can find clusters and classification, et cetera. Everybody with me here? So basically, you dissociate single cells, you do sequencing-based counting of RNA reads for every gene, then you have the counts for every cell, you have multiple samples and cells from every different condition. So you might have a bunch of Alzheimer's patients, and all of these reads you're going to call as Alzheimer's reads. And you can think of this as a flat model that simply says, oh, I get like that many cells from Alzheimer's patients, or as a hierarchical model. First, I sample a patient, then I sample cells from that patient, and so on and so forth. So that's sort of the hierarchical approach of seeing it. And then you can basically do clustering or classification. Everybody with me here? Awesome. So very simply, we're going to be measuring 20,000 genes in hundreds of conditions in thousands of cells, and we're going to be ending up with every condition where I did a measurement, I get the expression of a bunch of genes, sometimes partitioned by cell. And then for the next condition, the next condition, the next condition. This could be, I don't know, 400 individuals. And that basically gives me an expression profile for a given gene across these conditions. So I can start asking questions about the experiment dimension or about the gene dimension. Everybody with me here? So we can ask how similar are two genes in their expression patterns and how similar are two experiments in their expression patterns. Everybody with me here? Awesome. So now what's clustering, what's classification? Clustering is basically saying, okay, I have these genes by conditions matrix, and I'm just going to find common patterns. I'm going to find genes that behave similarly across conditions and conditions that behave similarly across genes. Everybody with me here? This is just about finding common structure. The goal of clustering is to group similar items that likely came from the same category, and in doing so, reveal hidden structure. Now, classification basically says, okay, well, we have some B cells and some, T cells and some activated B cells and some proliferation, cells. And then from that, we're going to now classify genes that are related to these conditions or conditions that are related to these genes and so on and so forth. So basically, it's when you have known classes and then you're finding features that predict those classes. So the goal is to first extract features from the data that best assign new elements to one or more well-defined classes. Sounds good? All right. So I'm showing you this like that, but in fact, it's more like this. Okay. Right now, we're thinking about, we have two dimensions, like measurement in brain, measurement in liver. And that's a toy example. There's like 22 cells here and 2 measurements each. In practice, this is what real world data looks like. This is 2.4 million cells across 430 donors, every one of which has a 20,000 dimensional vector. Okay, and going back to your comment, yes, you can represent this into dimensions using some kind of nonlinear dimensionality reduction technique, which we're going to talk about in later lectures. Everybody with me here? So basically, remember that it's much more complicated. But in this lecture, we're going to think about the simple scenario to introduce the machine learning components and the inference little by little. Okay? So basically, these clusters discover one component of the variation, which is the cell type. But now within that cell type, the cells here come from, let's say, Alzheimer's, and the cells here come from control. Or the cells here come from males, and the cells here come from female donors, and so on and so forth. Okay? And again, single cell analysis is a lot more complex. We're going to talk about that in lecture 6. You have to deal with batch effects. For example, most of the time when you do an experiment, you basically see, ooh, this is the cells from the first experiment, and this is the cell from the second experiment, and they have nothing to do with each other. So basically what you need to do is find some anchors that are sort of the best, neighbors, for example, between these cells and kind of like pull them together and then, change the dimensionality to match those, etc. So that's the first thing. You got to remove unwanted variation. You got to understand that there's thousands of genes measured by cell. There's like dropout events. There's a lot of noise and sparsity because the samples are tiny. You're measuring one cell at a time. There's heterogeneity. There's all kinds of scalability issues. You got to work with alignment and integration across platforms and different experimental designs. Sometimes the little bubble will catch 2 cells instead of 1 cell, and that's a problem. We call these doublets. Sometimes you have multiplets. And you have to worry about dimensionality reduction, which can be linear, sparse, non-linear, etc. Everybody with me here? So all of that will be in lecture 6. But for now, let's worry about beautiful data, 2 dimensions. Let's cluster them. Okay. So how do you discover these clusters? How do you find, that these are microglial cells and these are oligodendrocyte progenitor cells and so forth? The way you're going to do that is by assuming that there's some fixed number of clusters and then partitioning the points into those clusters. And how? You can initialize randomly K cluster centers. And then you can repeatedly assign points to a center and then move the centers to the center of gravity of their points. And you can stop when there's no more reassignments. Let's see what that looks like. So I've sampled some points without knowing which distribution generated each of them. I randomly throw 3 centers. Yeah, this one, I may have cheated. I've put it very close to those points. But this one is sort of exactly halfway between those clusters. So what's going to happen? Let's run the algorithm. First, I'm going to assign every data point to the nearest cluster. Then I'm going to color the points based on that nearest cluster. And then I'm going to recalculate the cluster centers. I'm going to move the clusters, center of gravity of the points that are assigned to those clusters. Notice what's happening here. The green point, the green centroid, gets moved somewhere to that, average. But because I had the purple point here capturing some of those, we'll see what happens at the next iteration. So here, I've now captured those and those. And now I'm going to move the green point, the green centroid, closer to the points that are near it. And the points that are near it now, the purple cluster that came down has now captured all of the points previously assigned to the green. And now the green gets moved to the center of the corresponding points. Everybody with me? So what did I do here? I basically just cooked up a recipe. This is like AI. I cooked up a recipe. I don't tell you if it's good. I don't tell you if it's bad. I just simply, run an algorithm. It's basically just a recipe, like the way you make waffles or crepes. This is just a recipe. Now, how do we know if it's any good? Well, we need to evaluate it. How do we know how well it captures the thing that we were trying to capture? We can have some kind of approximation bound that basically says, here's how good or how close you get to the optimum. Okay? But let's now break it down a little bit. And then we're going to show that it's in fact Gaussian mixture model. Okay? So in one step, we basically said we're going to assign points to the nearest center. And in the other one, we updated the center to match the points. Okay? And guess what? This is a form of learning because I'm actually getting progressively better at capturing this unknown structure of the data. It's not supervised learning, but it is a form of learning. Okay? What are we trying to do? What is this algorithm actually doing? What it's actually doing is that by reassigning the point to the nearest center, it's actually minimizing the distance between all of the points that are assigned and the centroids to which they're assigned. And when I'm updating the center to the mean, I'm basically, again, re-inferring something about my representation of the world. So first of all, I'm saying which clusters generate the points, and then I'm saying what is the most likely clusters. And if either of them was known, the other one would be trivial. But because neither of them is known, we're kind of starting with a random guess of the clusters, of the centroids, and then we're iterating to improve it. And that's going to be a common technique for many, many algorithms that we see, where you basically start with a random initialization and then you progressively make it better. Now, you can think of k-means as basically trying to create clusters that minimize a cost criterion associated with the size of the cluster. You can basically say that the cost of inferring some structure to all of these points is simply the sum over all centroids or clusters of the sum over all points in every cluster of the total distance. Does that make sense? So by making the clusters compact, I can basically make that cost smaller. What's a great way to make this cluster super compact? If I have 1,000 points, choose 1,000 clusters, right? Boom, every point gets a cluster. Zero cost function. So, we basically can't have that one single measurement as the cost function. We also have to think about generalization. You have to think about the compactness of the representation. And that's a lot of what we call regularization. Regularization basically means that I'm going to have a penalty associated with a number of parameters that I'm using. Everybody with me here? Great. But basically for a fixed number of clusters, then this is a fine, algorithm that basically minimizes the spread of these clusters. Sounds good? And it turns out that the k-means algorithm is basically minimizing this geometric view of the structure inference of the points, which is we're, trying to minimize that total sum of distances. And what is that sum of distances? Basically, the way to do that is simply the centroid of the points is what minimizes. Okay? So you can basically think of the algorithm as basically doing that. Now, assigning these points discretely and unambiguously to only one cluster at a time has a problem, which is that some points can truly be almost halfway between two centers, and therefore we might want to sort of think about partial weights. And that's going to be a modification of the k-means algorithm that's like fuzzy k-means. And what is fuzzy k-means? Fuzzy k-means basically says we're going to basically assign points partially to each of the clusters. Instead of saying this point is 100% blue, we say that this point is 98% blue and maybe 1% purple and 0.7% green. Everybody with me here? And this point, for this point, it probably doesn't make a big difference, but for this point, it does. It's like 40% this and 30% that and 20% this or something like that. Okay? 30%. Anyway, so the goal here is instead of saying that there's probability 1 of being generated by this cluster and this cluster only, we say that now it's a probability that really depends on that distribution of the points. So, and again, that's where we're getting to the Gaussian mixture model. Every single one of these centroids is a Gaussian generating points from this distribution, randomly sampling according to that Gaussian distribution. And now when you see these points, you say, okay, which Gaussian is more likely to have generated this point? Okay, so now let's think about the machine learning formulation of this nice little recipe that we saw. You basically have the generative model. Again, I have a model of the world and I'm sampling points from that model, as we saw earlier. So I have a model of the world that's, there's some red points and there's some blue points. So I flip a die and I choose, or flip a coin and I choose whether I'm going to sample from the red distribution. And when I do, I basically sample from a Gaussian. And therefore, the points are going to be distributed from the center out in an exponential decay. Okay? And this is the Gaussian distribution. It's basically 1 over 2 pi, and then that's the exponent, and that's the distance, and so on and so forth. So that's the decay of the Gaussian function. And the Gaussian function is not just something that humans dreamt up with to make high school students suffer. It's real. It's there. It's there in the real world. So basically, when you create a machine where every ball will randomly fall on a stick and then randomly go right or left with equal probability, you end up with basically a Gaussian distribution. And it's spooky how present and omnipresent it is. Okay? So then you can think about these centers of mass or the center of gravity, and you can think about the spread of that distribution. And now the question is, if I have Gaussians generating points with means, here, here, and here, and here, then what is the most likely Gaussian to have generated a point here? Well, the red one. What about here? Well, the orange one. So the rightmost point comes from the orange distribution. What about the leftmost point? Also the orange distribution. That's a little weird. And that's because it has the longest spread. And therefore, the fact that the green one is slightly to the left of it matters initially, but eventually stops mattering. Yeah.
So in the k-means algorithm, how do we know 1 point on the bottom left of the graph versus 1 point on the top right of that graph is not from the same distribution? But k-means would assume they're different distributions, right?
Exactly. So basically, now we're getting closer to fuzzy k-means and to expectation maximization. So basically, the original k-means, the geometric approach, only deals with one unit variance. It doesn't understand about different variances, and it doesn't understand about priors. So basically here, in the full model, we'll have different priors for blue versus green versus red versus orange, and different spreads. Whereas k-means only understands about the means, but not the standard deviations or the frequency. Okay. So now we can go back and revisit this cute little recipe and basically realize that it's actually an optimal algorithm for finding the interpretation of the world that has maximum a posteriori probability of generating the points that we observed. Is everybody with me here? So what we're going to do is reinterpret these 2 steps of the k-means algorithm in an expectation maximization way. What is expectation maximization? It's a way to iteratively refine the latent variable, for example, the centers of the points, and the rest of the distribution in an iterative fashion. Instead of saying, I infer one thing about my data, it's an iterative way to estimate an underlying posterior distribution that you don't directly have access to. Okay, so the E step assumes that the centers are known and it estimates the memberships. The M step assumes that the assignments are known and recomputes the centroids. Okay, and iterating between them, you basically have a set of label points. Oh, that's great, super easy. I can find the centers. If I have known centers, oh, super easy, I can assign the points. But the iteration between them is what basically allows me to get learning, machine learning out of this process. Okay, and then the solution to the model of the world that maximizes the total probability of generating these points is no other than the answer of the k-means algorithm. Basically, what you're trying to do is solve two problems at once. The first is infer which points were generated by which clusters. And the second one is, what are the clusters that generated the points? And you can infer both. If you knew any one of them, the other would be easy. But if you don't know either, then you start anywhere and you iterate. Okay? So then the maximization of this probability of generating the points given that Gaussian, again, it's a generative model. We have the forward probability of inferring the x's, which I'm sampling from the Gaussian centered at that point. That total probability, the argmax of the log probability, is basically the same as that probability is this expression here. And then this becomes, irrelevant because it doesn't vary with x. But this here is nothing but the solution to the centroid problem that we had already. Okay, yeah.
Yeah, I'm just a little bit confused because we define the method with Gaussian Newton model that also can infer variance of the cluster.
Correct.
Like a k-means.
Correct.
It's only log means.
That's exactly right. So basically, in the case of the variance being the same, it's identical. If the variance changes now, then then we need a more sophisticated version of that algorithm. Okay, so basically, when I'm computing that probability here is just simply based on this uniformly decaying function. But now, if you have a parameter associated with the spread of that decay, that becomes a standard deviation, and that you scale in that exponent, and that basically becomes a scaling factor that you're going to need to incorporate in your algorithm. Okay. And then again, the same thing happens with this maximization of either maximizing the likelihoods of the cluster means and maximize the likelihood of a point being generated by that mean. And you can basically think, again, side by side about the k-means algorithm or the fuzzy k-means algorithm and what is the E step and the M step. And, I'll let you, walk through that on your own. But the initial k-means solution was a uniform prior, where every cluster is the same size, a unit distance function instead of having some variable standard deviation for your points, a symmetric distribution between the different dimensions of your data, where every dimension was independent, the x and the y were independent, whereas here we can actually talk about a covariance matrix of sort of some, reshaping of the space. And then with k-means, we simply pick the max, where we found the one cluster that was closest, and we chose that one. And with fuzzy k-means, we looked at the full density. And with expectation maximization, we can look at the full density and then estimate the membership of the points all probabilistically according to each cluster. Or we could even sample from that posterior distribution using Gibbs sampling that we're going to see again in a lecture or two. Okay. So again, if you have an original data set that was generated by a big green cluster that was, shaped this way, and then two little red and blue clusters, the k-means solution will spread them, exactly that way. We'll sort of separate the points that way. We'll partition, I should say. Whereas the full EM model will be able to sort of recognize the standard deviation of the blue is actually larger than the standard deviation of the green, and so on and so forth. And therefore, that point here, even though it's technically closer here, will be assigned there. Okay. Excited? Good. So that's basically expectation maximization. And that sort of assumes that we have chosen a number of clusters, and then we're partitioning into those K clusters. Okay. But the problem is that selecting K can be hard. You can always make clusters more compact by increasing K. And if K is the number of data, then, of course, it's, perfect, but meaningless completely. And with hierarchical clustering, you can kind of sidestep this. So basically, hierarchical clustering is an agglomerative clustering that basically says, can I build a nested set of clusters which are organized as a hierarchy? So when I have a set of points, I don't need to choose K in advance. I just find the two closest points and I merge those. I find then, then they become a cluster. Then I find the next closest there and I merge the next two closest clusters. And I continue to basically generate a tree, a hierarchy that basically tells me the order in which they were clustered. Okay. And then if I want to make a cut, I can just choose how many clusters do I want. And I can just make a cut at that height. And then it gives me the 4 clusters. Everybody with here? Good. So this is UPGMA that we're going to see again in phylogeny later in the semester, which is unweighted pair group method with arithmetic mean. And when you do that, you assume that every point is a cluster initially. And when you merge 2 points together, you merge the corresponding clusters. But the problem is that the cluster-to-cluster distance is itself a matter of debate. How do you calculate the distance between this cluster and that cluster? You could take the distance of the closest 2 points. You could take the distance of the furthest 2 points. And if you choose this one versus that one, this one will basically allow you to merge clusters like a snake, connect them together, whereas this one will sort of tend to make clusters that are very compact because it wants to make sure that the 2 furthest points are no further than any other cluster that I could merge. Is everybody with me here? Or you could say, let me take all of the pairwise distances and just average them, and then that will be the cluster-to-cluster distance. Or I could simply take the center of gravity and the center of gravity and, look at the distance between the centroids of those. This one is a k-squared algorithm. This is an n-squared algorithm. This is basically checking all n-squared distances. This is only checking all k-squared distance, where k is the number of clusters at that point. But of course, recomputing those distances basically means that I need to, now that I've merged these clusters, I need to recompute the center of gravity. Okay. So anyway, basically the cluster distance affects both the results and the runtime. And again, the point-to-point similarity can be either Euclidean distance, which is what most of us intuitively think of, or it could be Manhattan distance, or it could be Pearson correlation or Spellman correlation, and so on and so forth. Okay. And then to evaluate the clustering results, you can basically look at some other independent measurement and say, how likely is it to have sampled, I don't know, K Alzheimer's patients in my group randomly by chance. Okay. So I want to make a small parenthesis here because we are in 2024 and people think of clustering of points with numbers, but you can actually cluster things beyond just points. Okay. So here's, of course, clustering of numbers. So basically this is, 2.4 million cells clustered according to their expression patterns. This is now patients clustered together, but you can actually start clustering text. How do you cluster text? You can cluster text by, and I don't know if that link will work, but let's find out. Yoo-hoo. So this is basically clustering of responses to a survey. So basically this person says, oh, I have a relationship that did not go as expected. As a result, it brought me down personally as a person. I'm sorry, this is very depressing. So as soon as I said depressing, all of you guys are laughing. Okay. So basically, back in 1980, a bunch of old men sat together in a room and said, okay, we're going to define psychiatric conditions according to this list. And now every one of us in 2024 has to fit one of those lists that they decided like 30, 40 years ago. Okay. Instead, you could say, okay, let me just look at the responses of every person. And then based on that, I can infer a hierarchy of, psychiatric, conditions. Basically, all of these are parental health concerns. Okay, this is in Greece, so it's a little biased. Personal concern summary, etc. So basically, relationship breakups. So all of these things, it happens to you. That's the thing. Like you suddenly realize that it's not just me. I'm not the only weird person who doesn't fit one of those like 1980 categories. Like all of us are struggling with some of those things. So you can basically now, study not just numbers, but also text. You can take the description of every single human protein, just as free text. And then you could cluster those descriptions together, and hopefully that space will come up. And now you can basically navigate 20,000 genes, not based on their expression, but based on the description of their functions. And if I search here for lipids, for example, if I can spell, I can basically find that there are, genes associated with lipids in a bunch of different categories, maybe in cargo receptors or in biosynthesis and molecular function, etc. You could also do all kinds of fun things. You could select those points together and say, what are some, let me make this a little bigger, diseases that these genes are involved in, right? And then it can basically, tell you answers about that. Anyway, why am I showing you this? Because this is so much fun. A bunch of undergrads, like mostly freshmen and sophomores in my group, built this. And it allows you to now start thinking of text as clusters. So this is another fun space. Every Lex Friedman clip is now categorized. And you can see here, unsurprisingly, when you zoom into the center, that's where the meaning of life is. And next to meaning of life, you basically have philosophical discussions, etc. Why am I showing you these? Because I want you guys to think broadly beyond just numbers. Everybody's thinking of, I'm going to do a final project by measuring expression here and here and there. Why don't you just do a co-embedding of, all of the expression patterns of, a bunch of genes with the latent embedding representation of the corresponding things, okay? So that's the sort of small parenthesis. And all of this to say that what we just saw is a much harder version of naive Bayes classification. Why? Because with naive Bayes, you have classes that you already know about. You have class 1 and class 2. And therefore, you can assign probability distributions to the points from that class. So before, I was doing everything from scratch. I didn't know what point belonged where, and I didn't know what the cluster would look like. Whereas here, you actually know what points belong where. And therefore, it's actually super trivial to infer the center of mass of that distribution. It's just the average of those points. The spread is just the standard deviation. And you can now use that to start inferring what is the probability of a given class given the data. And how much more likely is it that something belongs in class 1 versus class 2 given the data. And of course, as we mentioned earlier, the denominator is the same. So what do you want to know? You want to know the probability of class 1 given the data, probability of class 2 given the data. And using Bayes' rule, this is the backward facing arrow. This is the forward facing arrow. I know how likely it is to generate a particular expression value given the class. And I know the prior for that class. And the denominator is the same. So I can basically build a Bayesian classifier between different classes simply based on the generation probability times the prior. A lot of people think of Bayesian statistics, oh, you just have priors for everything. No, the prior just simply comes from this inversing the direction of the arrows part. And the prior just naturally sort of falls in there. And therefore, we can think of a discriminant function for a classification based on whenever that probability is greater than 1, or whenever the log probability is greater than 2, then we classify according to one class or another class, okay? And therefore, we can think about estimating these distributions based on the number of points that are sitting in different parts. You could assume a parameterized model, which basically means I have a mean and a standard deviation. Or you could assume a non-parametric model that simply says, I'm just going to break up the distribution in different parts and just simply measure how much is in each part. If you know that your data is distributed as a Gaussian, you're doing great. You can just use that. If you don't know, then you want to be a little more unbiased. For example, it could be a multi-peak distribution or a non bell-shaped distribution, and so on and so forth. Is everybody with me here? Awesome. So, and then to combine multiple features, that's where you can assume in the naive Bayes scenario, you just assume independence. You assume that all of these features are independent of each other. And that's the naive Bayes assumption. It basically assumes that all of the features are independent, and therefore that the joint probability of all the features together given a class is simply each of the features in isolation, and therefore the product of a bunch of independent classes. Of course, this is almost never the case. However, you could do some kind of principal component analysis. You could basically separate all of these different measurements into a set of separate principal components, and then you can actually use, Bayes classification that way. Okay. So again, this was optional, so we're not going to cover it. But basically, we talked about gene expression analysis. We talked about k-means. We talked about hierarchical clustering. We talked about naive Bayes classification. And we also talked about how this does not apply solely to numbers. It can apply to text. And you can really go wild because it is 2024 and, large language models are around. And sort of what I showed you earlier with the text classification, this is super trivial. It's basically just looking at the latent embedding representation of a large language model parsing of whatever text it is. So you can basically, use that. And if you guys want to use it for your final projects, I would love for all of you guys to be able to visualize, to sort of zoom in to the different clusters that you're learning and so on and so forth. Okay. So that's where we end for today. And I will see you guys on Thursday, where we're talking about some more fun stuff. So on Thursday, we're going to talk about hidden Markov models, dynamic programming, parsing, and sequential data. Yeah. So hold on, let me stop the recording.